Hi everyone! This is me, Teacher John, and for today's video, we will discuss about DNA replication in prokaryotes. Are you ready? Let's begin! DNA replication will begin to a specific area of the molecule called the origin of replication. The ORC denotes the area of active replication called the replication fork. The helicase separates the strands of the double helix, while the single-stranded binding proteins stabilizes the newly single-stranded regions. DNA gyrase or topoisomerase is used to make sure that the double-stranded areas outside the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Short sequence of RNA called primers have to pair to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. There. Now, the DNA polymerase is ready to add new nucleotides to the growing daughter strand. As it passes through, it will add new strands of nucleotides. Replication of both strands occur at the same time. Here in our experiment, one uses continuous synthesis and the other discontinuous. Continuous synthesis here in the experiment occurs in the 3' to the 5' oriented parent strand, which is referred to as the leading strand. Now, new nucleotides are added to the 3' end, moving continuously towards the expanding replication fork. Discontinuous synthesis, on the other hand, occurs in the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in the segments called Okazaki fragments. Now, DNA polymerase, with the use, of course, of primase to add primers ahead to the 5' end of the lagging strand, which is this one, there. Now, the DNA polymerase 3 then adds short sequence of nucleotides, here. Polymerase 3 added short sequence of nucleotides, the Okazaki fragments to the primer, filling in the gaps. As the helix is opened further, this process repeats until the entire strand is replicated. We have here the DNA polymerase 1, which replaces the RNA primers with DNA nucleotides, here changing the uracil to thymine. DNA ligase, in addition, is used to ensure bonding between the fragments, which is the Okazaki fragments, and replace nucleotides. Say, for example, a while ago, we have here a gap. So, the DNA ligase is responsible to fill in the gaps in order for the sequence to be completed. Once the leading and the lagging strand have completed the replication, two identical copies of the DNA molecule results.